In this video, we'll just spend a few minutes and talk about cell junctions. Now, often in tissues, cells are not adjoining each other, but in many tissues, they are. Examples would be in the upper layers of the skin, the epidermis. Also, when we start to look at things like heart muscle, skeletal muscle, and a whole number of other tissues, the cells are pretty well in contact with each other. So we should talk a bit about the different types of cell junctions. And really, this comes down to how do cells bind with one another. Now, one of the big things that we see is that, remember cell membranes are full of proteins, different types of proteins with different functions. But neighboring cells, plasma membranes, are linked by those integral proteins. And there's three main configurations we can see. We have tight junctions, we have desmosomes, and we have gap junctions. And structurally and functionally, they're very different from each other. And let's have a look. So when we're looking at tight junctions, I prefer this name versus occludent. Tight just tells us that that there's no wiggle room between it, right? Between the cells. These hold cells closely together, and the space is impermeable to the movement of macromolecules. So when we're looking at tight junctions, you can see that here they are. Is that they're so strong, so they run all the way around, that they really prevent materials from sneaking in between cells. They just don't allow that to happen. If you want sort of an analogy, think of like Ziploc baggies. The part that you zip together, that's sort of the idea of a tight junction, that it's not just a one little space, it's a continuous along the margins of where the cells interact. So, as I said, it's basically impermeable to the movement of macromolecules. And what we have is the integral proteins of adjacent cell membranes are locked together to form a seal around the apical perimeter of the cell. Apical is the top bit, and basilar would be down at the base. Now, the seal may not be complete, allowing for leakage in some tissues, but in many tissues, it's a very efficient way to prevent things to sneak between the cells. And one example would be between cells and blood vessels, and this prevents blood cells from basically leaving the blood vessels. So, and there's a number of cases. Really, where do we see tight junctions? It's when I really want to separate this area from this area, and I want to be able to control molecules moving back and forth. Now, by having a tight junction, it means the only path for these to get through is often enter a cell, cross it, and leave it, which would be transcytosis. So the cell has much more control as to how things are moving from one side to the next. Now let's look at desmosomes. Now desmosomes are these linking integral proteins, and they do allow for materials in the extracellular fluid to pass through the space between the cells. So here's a desmosome right here. And you can see that here's one cell's membrane, here's the other. So you can see that we have quite a structure. We have these intermediate filaments. We have this plaque area here, and then we have a number of cell adhesion molecules that are sticking out. And think of it almost like a little Velcro area. Now, up here, you can see tight junctions go right around the periphery of the cell where they interact, whereas desmosomes are only little patches. So there'd be one here, but between these two cells, there wouldn't be necessarily one there. So material could basically get around that way. Now, where do we see these? Now, the one thing is that they have a fair bit of strength. So the integral linking proteins are attached to the intermediate filaments of the cytoskeleton. And desmosomes are really about structural reinforcement to make sure cells do not get pulled apart. And so really, wherever we have tissue with a lot of mechanical stress, and a great example is the epithelia of the skin, we will have desmosomes there. So they don't prevent things, as I said, from getting between cells, from one side to the next, and squiggling between the two cells. What they do is they really assure that the two cells don't get pulled apart. And the next one are gap junctions, and the name gives you a big clue that they do have little gaps or small pores that are formed by protein channels between the adjacent cells. And this allows for certain substances, and of course they're really small substances, to flow between one cell cytoplasm and the others. And one great example of this would be electrical signals in the heart muscle and the cardiac muscle. And when we get to the heart, we'll come back and talk about this. So really, gap junctions really have a role in cell-to-cell communication. And let's just kind of zoom in here a bit. You can see that here's a gap junction. You can see that these are the protein channels that form. This is one cell's membrane. This is the other. There's a channel that's formed here, a channel that's formed here, and that allows for the passage of things such as really small stuff like ions and things like that. So those are the three main types, and here they're just sort of shown once again. So let's just quickly sort of reinforce our knowledge. Remember, reviewing or repeating is a good way to do it. When we look at cell junctions, we have tight junctions, we have desmosomes, we have gap junctions. Each one of these is for a different function. Tight junctions, they really run all the way around where the cells meet. Think of it like that Ziploc baggie, right? Desmosomes are about dealing with mechanical stress, and they're just in little patches. They're in little patches that would be where the membranes attach to each other, and gap junctions allow for small things to move back and forth. Now, once again, make sure you go into your textbook, you read the corresponding pages. It'll talk you through it a bit more, but I want to at least give you a sense of the basics, and so you walk away with knowing the three types, their names, and really what their function is, and get a visual as to what they look like. Okay, we're going to end the video there.